All right, we're talking about population genetics today. So what we've been talking about so far in the semester has been how are alleles passed down in generations, right? So we've been talking about traditional genetics, Mendelian genetics. We've been talking about how the DNA is modified in the cell. So we've been talking about cytogenetics or epigenetics. I want to talk now about population genetics. Right? Instead of the genotype of the individual and ancestors or the individual and their offspring, I want to talk about the individual or the genotypes that are present in an entire population of organisms that may or may not be breeding with each other. So questions in population genetics, and this is relevant for if you're studying populations of birds, populations of organisms, populations of people. Often if you're doing disease research of how is a certain disease affecting a certain region of the planet, then you'll do population genetics. Is there a background genetics that's affecting how this disease is being spread amongst people in a town, in a village, in a city? So I'm going to do some really basic stuff, and in fact, most of this probably has been reviewed for you. You've probably seen it at some point or another, probably in Gen Bio. We're going to talk just a little bit about calculations, about how you would go about estimating what the frequency of certain alleles and what the frequency of certain genotypes would be in a population. Okay. So how widespread are certain alleles in a population? And how do allelic frequencies in a population change over time? Are the big questions in population genetics. So in our examples, we're just going to talk about two alleles. The math holds the same, but it just gets more complicated when you're talking about multiple alleles. So obviously, we're talking about a very limited example here. And the math is going to be very limited to only two alleles. For most human traits, you're going to have multiple alleles in a population, and so it becomes harder to keep track of them. But for the simplicity of the math and just to show you what the actual theory behind this is, we're just going to limit ourselves to two alleles. Okay? If you're talking about a certain disease, and you're talking about an autosomal disease, and it would be homozygous recessive, we're talking about a recessive allele, then this math would actually probably work for you, because you could just say, well, the little a is the diseased allele, and any other allele, I don't care what it is, isn't going to give you the disease. Then you can kind of boil the math down to this. But if you're talking about a complex trait that's got multiple alleles, having different phenotypes associated with them, then this isn't going to work. So in a population, if we have a gene that's got only two alleles, the frequency of the wild type or the dominant allele, we call P, okay? So that's just how often, it's a percentage number, right? How often is that allele found in the total number of alleles, right? We're talking about alleles at this point, not genotypes. And then the frequency that you would find the allele A, we call Q. If these are the only two alleles in the population, then the frequency of P and the frequency of Q equals 100%, right? So, what's that? Well, if there's only two alleles in the population, then the frequency of the dominant plus the frequency of the recessive equals 100%. Or if we're going to use fractions, we'll just say, point something plus point something equals 1. Right? This formula and this way of thinking actually developed really early in the history of genetics when Bateson and Punnett and these guys were repopularizing Mendel's views. Right? We discovered that Mendel's laws were true of not just peas, but of organisms in general. There was criticism originally because of this idea of dominant and recessive traits. The idea was, well, if a trait is dominant, then it should just be the most widespread trait, right? A population would just tend to always acquire and keep the dominant trait. So this was a problem when people were initially thinking about it. That kind of, in some ways, makes intuitive sense, right? If it's the dominant allele, then it should just dominate the population and just spread, and everyone eventually should just, as that population reproduces and the older generation dies off and the new generation comes up, the dominant allele should just be the most prevalent, right? It was a guy named Hardy. So Reginald Punnett gave this problem to a guy named, what was his first name? It was like Gilbert. Godfrey Hardy was the guy who worked on this. And so this was called Hardy's law for a long time. And Hardy's law says, whatever the starting frequency of alleles are in a population, dominant alleles are not going to take over, recessive alleles are not going to go away. Hardy's law says if a population is an ideal population, the original allelic frequencies should remain the same over time. So this is called Hardy's law. So the frequency of alleles in a population should, maybe I should have put should instead of will, should remain the same if the following 7 conditions are met. So you started thinking about populations. What would have to go on in a population for one allele to become dominant or the other one to disappear, or for alleles to stay the same? So these are the classic 7 conditions of Hardy-Weinberg population genetics. Weinberg was a German geneticist and population geneticist. He came up with these basic same laws independently. So Hardy and Weinberg didn't really talk to each other when they were developing this, but they both ended up coming to the same conclusions. So Hardy's law became Hardy-Weinberg. All right, so the first condition, mutation is not occurring. If the allelic frequencies are going to stay the same, then we can't have new alleles popping up, because obviously that just by definition is going to change your allelic frequencies. If you had 2 alleles, a mutation occurred, now I have 3 alleles, obviously the allelic frequencies have changed, right? So mutation can't be occurring. Number 2, natural selection is not occurring. That is, one allele is not giving any organism an advantage in surviving or reproducing. If one of the alleles is giving an advantage, then it is going to is going to start dominating, right? Because if people who don't have that allele are dying off more rapidly or unable to reproduce, then it's going to shift towards the favored allele, right? So if mutation and natural selection, if neither of those are occurring, then the allelic frequencies should stay the same. Additionally, the population is infinitely large. So obviously these are ideal circumstances, right? Mutations do occur. Sometimes alleles give an advantage in certain conditions. And there's no population that's infinitely large. I mean, maybe if you're growing a huge thing of bacteria, just got a huge tank of it, and I have millions, trillions of cells going on, maybe we start approaching Hardy-Weinberg ideal circumstances there. But in any normal animal that you're probably interested in, it's not going to be infinitely large, right? Number 4, all members of the population breed. Now that's not true either, right? We have some ages of the population, sometimes certain people are reproducing, and you might still be a part of the population, but you might have gotten to the point in which you're not breeding anymore. So if all members of the population are breeding, that means that equal exchange of allele is just as likely, right? One individual is just as likely to breed with this individual as any other individual in the whole population, right? So for the statistical math to work, the alleles all have to have basically the same ability to interact with each other. And so that says all of the members must be able to breed, and all the mating is totally random. There's no preference for alleles, there's no sexual selection, there's no nothing going on, right? These are obviously ideal circumstances. So anytime you're using Hardy-Weinberg, you're always making an approximation. And the closer your population is to these laws, the better your approximation is going to be, but it is always an approximation, okay? Number 6, everyone produces the same number of offspring. That's not true. But some populations more or less are like that. Armadillos are weird. Armadillos, when the female has a fertilized egg in her womb, it grows to the 4-cell stage, and at the 4-cell stage, all 4 cells disperse, and almost every armadillo breed is 4 identical offspring. So I guess the armadillo is the closest one to being everyone produces the same number of offspring. But You know, some animals have big broods and they, you know, give lots of offspring. Sea squirts just spawn their eggs into the ocean and could have hundreds of offspring every morning. Populations differ, right? Number 7, there is no migration in or out of the population, right? You can think of populations as like the entire population of every, I don't know, armadillo on the face of the planet. If you're thinking about that as your population, then obviously you've met number 7, right? There's no migration in or out of the population unless you're getting extraterrestrial armadillos. That's probably unlikely. But if you've met number 7 by saying the entire population of these organisms on the Earth is your population, then you're probably not producing the same number of offspring. Mating is not random because, you know, armadillos on one side of the country are not mating with armadillos on the other side of the country. Probably not all members of the population are being able to breed. So sometimes getting closer to one of these ideals means you're getting further away at another one, right? So no population ever meets these. Questions about the conditions? All of these have to be met for the math to be a good approximation, right? Even if all this was true, you're still an approximation, right? Yeah?
Clearly this supposed to work well enough to still be used. So is the approximation...
It's better than nothing, right? Yeah. So, I mean, math is always idyllic, right? Unless you're building in multivariable calculus, right? We're not doing multivariable calculus, but the math here is making assumptions. So what we do is we just make it as clear as we can what we're assuming, and then just estimate your, deal with how close your estimation is based on how close you think you are to these conditions, right? So it's better than nothing. If you're going to try and get an idea of how often an allele is, you can't genotype everybody in the population, right? I mean, you could always get around population genetics math by just going and sequencing every individual, but that's just not practical, right? So aside from that, we just do the best you can. All right, let's take a really simple example here. Here's a little population of pigs. Pigs on a farm. I got 2 phenotypes here. I've got pink pigs and I've got black pigs. The pink pigs are the dominant wild type. The black pigs are carrying a homozygous copy of a recessive gene, okay? So the dominant trait is pink. This is the phenotype. The genotype underlying pink could either be homozygous dominant or it could be heterozygous, right? Because the big A gives you the pink coat color. So I've got 2 genotypes possible to give me pink, right? The homozygous recessive is, we'll call it little a. That's going to give me the black phenotype, okay? So if I look at my population, this is the extension of Hardy-Weinberg then. Okay, so if the frequency of big A is P, right, the frequency of the allele big A, that frequency is P. The frequency of this genotype, having 2 A's, is P squared. Okay, so when you're doing population genetics, make sure you keep clear in your head. What am I talking about? Am I talking about the frequency of the allele in the population, or am I talking about the frequency of the genotype? The genotype is always 2 alleles, so you have to do P squared for the homozygous dominant. For the homozygous recessive, we call that Q squared, right? The frequency of small a would be Q, 2 small a is Q squared. The frequency of the heterozygote is the frequency of big A and the frequency of small a, PQ, P times Q. But there's 2 ways to get that, right? You can get a homozygous or a heterozygous by inheriting big A from mom, or you could have inherited big A from the dad, right? There's always, in a Punnett square, there's always 2 ways you can get the heterozygote, right? So this is 2 times P times Q, okay? That's the probability of finding a heterozygote in this population. So the math works out, again, talking about the total population. The total population has to be one of these 3 genotypes. And so the frequency of the genotypes is p squared for the homozygous dominant plus 2pq for the heterozygotes plus q squared equals 1, equals 100%. Does that make sense? Sure, yeah. Okay, so how do we analyze this population of pigs? The easiest thing to do is identify whatever it is in your population, identify the individuals that have to be just one of the 3 genotypes. Okay? It wouldn't be helpful to count pink pigs right now, because pink pigs could be homozygous dominant or they could be heterozygous, right? But the black pigs are homozygous recessive, so you count the black pigs. To be black, you can only have one genotype, right? So we've got 16 total pigs here, and I've got 4 black pigs. So you can set up your math. You can say, in my population, I've got 4 out of 16 are black pigs, right? This is, so 4 divided by 16, 0.25. That is the frequency of the homozygous recessive. Right? So q squared, the frequency of homozygous recessive, q squared equals 0.25. You just solve for q. Q equals 0.5. All right, take the square root. Once you know the frequency of q, then you automatically know the frequency of p, because p plus q equals 1. Right? So you do simple math, p equals 0.5. Right? This is a very simple, very idyllic population here. Right? Once you know p and once you know q, now you can estimate what the actual frequency of homozygous dominants and heterozygotes are in this population. Right? So if you start with what you know, start with the black, who are the known phenotypes. The blacks are homozygous recessive. If you start with those to get your frequencies, then you can go back and estimate how many of these pink pigs are homozygous dominant and how many of these pink pigs are heterozygotes. Right? So you just plug it into to Hardy-Weinberg, or just, frequency of homozygous dominant pink pigs, 0.5 times 0.5, same as the frequency of homozygous recessives, right? So 4 out of these 16 pink pigs, or 4 out of the 4 out of the 16 are homozygous dominant, right? 8 pigs, so that would be 4 out of the 16. Heterozygous would be 2 times 0.5 q times 0.5 p equals 0.5. 8 pigs out of this population are heterozygotes, you estimate. Now, obviously, this is not meeting any of the Hardy-Weinberg principles, though, right? It's not an infinitely large. There might not be selective breeding. Maybe, having a certain coat color is disadvantageous in terms of finding a mate or surviving. These are out in a field. You can't tell if they're on a farm, so I don't know if there's any selection, artificial or natural selection going on. Maybe the farmer has actually been selecting for pink pigs and not allowing the black ones to breed, right? So obviously, my ratios might be off then, right? Maybe other pigs are coming into this population and breeding with these pigs, right? I don't know. So, since this is ideal, let's talk about what could actually exist in nature that would cause you to have deviations from Hardy-Weinberg. Although, should I stop? Or are there questions about how to do the math? You've been doing homework on these problems already. Are there issues that have come up? So speaking of, on your homework, there's one that has 3 alleles. If you know just how to set up the equation, don't worry about the multiple substitution algebra for it. I don't care that you actually do all the algebra, but I want you to see how much more complicated the equation gets if you throw in a 3rd allele, right? So if you can set up that first problem, you don't have to solve the problem. Okay.
So you're saying that will be on the test?
That will not be on the test. Yeah. I don't want you to spend all your time doing multi-substitution algebra. I'd rather you focus on genetics. So
the actual formula is like for the total population, it's like P2 plus 2.
yeah.
RP squared.
Yeah, so these should work, right? If you put in 0.5 and 0.5 every place here, that all should give you 100%. Right? Yeah? Okay, let's talk about deviations then. If any of the 7 ideal circumstances aren't being met, then the Hardy-Weinberg equation is not going to give you proper estimates, right? So what are these kind of specific events that could actually get you to deviate? We're going to talk about the first 4 in detail, so I'll just talk, and I've already kind of talked about mutation and natural selection, right? If there's new alleles popping up, then obviously this isn't going to work. And if there's some type of preference, either in the farmer breeding your population or something naturally occurring that's killing off certain genotypes, then that's not going to go. So I'm going to talk about these specific events. Number 1, genetic drift. Number 2, the bottleneck effect, which is related to the idea of a founder effect. And then 4th, I'm going to talk about gene flow. So I'm just going to step through these 4, and then we'll be done with population genetics. Genetic drift. Genetic drift happens in populations that are too small. If you think about a big population, by random chance, sometimes an allele might just randomly start to be preferred, right? Just because when chromosomes are segregating and eggs are getting fertilized, not every egg gets fertilized, and chromosomes can segregate independently. So just by random chance, you can sometimes get more individuals with, say, the dominant allele. But just by random chance, you could also sometimes get the population preferring the recessive allele, right? And under normal circumstances, in a really big population, you would see just kind of noise if you were keeping track of the allelic frequencies. Sometimes you'd see the big A allele get more preferred, and so on this chart, oh, it's pretty fuzzy. This is just the percentage of the allele, right? So this is 0%, that allele does not exist in the population, or 100%, that's the only allele in the population. If you started out with a ratio of 0.5 and 0.5, 50-50 chance of getting the allele, and as that population breeds over time, so in this chart, this is over time, you could have one allele start to get, it starts off as 50-50, but then just by random chance, it might, stagger up, and one allele becomes more frequent, and the corresponding allele becomes less frequent. But then over time, it's just random chance. Maybe it pops up for a while, but then just by random chance, it goes down for a while, and pops up for a while, and goes down for a while. This is just normal noise that would go on in a background, right? Now, if you have a big population, that noise kind of gets buffered, right? There's lots of people, if this one female has a lot of offspring that just happened by chance to have a lot of the dominant allele, then there's probably some other female next generation that's going to have a little bit more of the recessive allele. So it just buffers itself out, right? Because these are just random chances. The more chances you got, the more random it becomes, right? So a large population will be able to buffer that effect. If you have a really small population, though, if you have a population of only 16 pigs, one of those females, if she has all dominant alleles, that could actually drastically change because you only have 20 individuals in the first place. If you got 16 individuals, one female has 8 individuals, 8 offspring. What those offspring are contribute a lot to the overall population, right? She has 8 offspring, and we only started with 16. So whatever she had is going to contribute a significant amount to our allelic differences. So if by random chance she has a lot of dominant alleles, then I've drastically changed my combination in that next generation. And that's going to affect the subsequent generation, right? Because more often now, individuals are probably going to have that dominant allele. So here's two populations. And I think this was just a computer simulation of breeding over time. But here I've got a population of individuals. Here is a red. This red indicates one allele. And we'll say that this orange represents the other. So the red would be like the homozygous dominant. The orange is the homozygous recessive. When the dominant becomes more popular, when it becomes higher in frequency, then the orange one is going to go down in frequency, right? And if this is a small population, that could, just by random chance, take over the entire population, right? That one came up, and if it became the most dominant, if it became 100% of the organisms in that population have that allele, then nobody in the population has the orange allele, right? Those are inversely related. This is called genetic drift, right? I have just by random chance drifted and changed my entire population to only have one allele. Okay, so there's no selection going on, there's no formula to figure this out, it's just genetic drift. It's just by random chances, it just happened to do that. Now in a big population, so this is a population of 20 individuals, in a population with 20 individuals, you're gonna see genetic drift and you're gonna get alleles that become what we call fixed. A fixed allele is where it's the only allele in the population. In a bigger population, you'll buffer against that, right? So here, the red goes up and conversely the orange goes down, but then some other time the red's gonna go down, the orange is gonna come back up, and they're just gonna buffer each other. And you'll just see normal fluctuation noise in the background, right? Yeah?
So like, besides possibly changing like the phenotype of it, how does it affect the individual or like who has that?
Well, genetic drift is saying everything in Hardy-Weinberg is true, except it's a small population,
Okay.
right? So you would say there's no mutation occurring, there's no selection.
Oh, so it's not like changing the reproductive.
Nobody's getting, no one's preferring, right? There was not some selective event that said the red allele is the better allele to have. This is just by random chance, we just happen to lose the other one, right? Because they were 50-50 to begin with, and by random chance, this one jumped up. Well, now the population is not 50-50 anymore. It's preferentially the dominant one. So in the next generations, as we go along, that's going to preferentially take over, right? So only works in small populations. So that's where it's deviating from Hardy-Weinberg, is that it's too small a population. Okay. So nobody's changing phenotype here. All the phenotypes should be the same, or at least not being selected against, right? Maybe I lose black pigs, so that actually could be the case. I might be losing black pigs, so the phenotype might change, but it's not changing because being black was disadvantageous or being pink was more advantageous, right? It's just a random event. Right? Yeah.
So I'm trying to understand, so genetic drift is when the population is too small, and so as a result of chance, the alleles change to where one becomes completely
Fixed.
dominant or completely taking over the entire phenotype?
Yeah, so genetic drift just says by random chance, one allele became fixed and all the other alleles became zero. It's more often going to happen in a small population. A bigger population will buffer against it. Now, sometimes even in big populations, genetic drift happens, but it's less likely. Yeah. The bottleneck effect. The bottleneck effect says also more likely to happen in a small population. If you have a small population, then random instances are going to have a big effect on you, right? So if just randomly the offspring more likely came out with one of the alleles, that's going to have a big effect because it's contributing. The offspring are contributing in a substantially more significant manner if you're a small population than if you're in a large, right? In a bottleneck event, it's when a small population undergoes a catastrophic or drastic reduction of individuals, okay? This is just a random event. You know, I've got my 16 pigs and lightning strikes and kills off 8 of my pigs, right? For a population of 16, losing 8 of them is a big, significant deal, right? And if all 8 of them happen to be the black pigs, in those 4 that died, they were all the black pigs for whatever random reason, then my population is going to look very different. But not because of any selection, right? Just because small populations are will be heavily impacted by people being lost out of the population. Right? So if I started off, these are my allelic frequencies. So this is just an illustration to make you think of, I've got a population. In that population are a bunch of alleles, right? All right. So these dots are the alleles, not actually individuals. But if I have a population and by a catastrophic event, a bunch of people die off, starvation or lightning or, I don't know, just some major catastrophic change to their environment that just kills people, well, then I might actually get a small population. And if everybody died off except these individuals, then their alleles are going to contribute much more substantially to the new population when it regrows, right? I will have severely switched and biased my population to a certain allele, and now when that population starts breeding and reproducing, I've completely changed what the ratios were, right? So it's going to be pretty rare in a bottleneck that you took the same allelic frequencies and after the catastrophe you had the exact same frequencies. That's probably rare. Most of the time it's going to bias to one or the other alleles. Right. The same phenomena in terms of the allelic frequencies happens in a situation that we call the founder effect. The founder effect is, instead of a big catastrophe killing off people, a small enclave got separated from the rest of the population. Right? I got my pigs in a field. There was a flood. I created a river because of all this runoff. Some pigs are on one side of the river and other pigs are on the other side of the river. Now I basically have got 2 populations. And it was a random founder effect, right? Whatever the random pigs that just happened to have been on this one side of the field have now become the new population frequencies, right? So a founder effect is when a small group either migrates off or just somehow is physically separated from the rest of the group, and they now establish a new population. So the same thing's going to happen. Instead of dying off, though, I just have migration away, and I've got a small new population arising. Okay? So both of these are random events, right? I guess they're not random. I guess if you're a breeder and you quarantine certain animals, you can do selective breeding here. But we're trying to think of just natural population changes, natural environmental changes.
And these are all individual effects too. So wouldn't we most likely also experience genetic drift as well?
Well, exactly, right. So if you had a 50-50 ratio of alleles, but then by a bottleneck event or by a founder effect, you now have a much smaller population with a different allelic frequency, then you're much more susceptible to genetic drift fixing one of these alleles, right? And in this case, that's what I'm showing you. This population, the only people that survived have the recessive allele, right? So that, you can have fixation on an allele because of a bottleneck. You can have fixation of an allele because of genetic drift, right? And they do kind of add on top of each other. Right? Last one. Gene flow. This is hard to get a hold on if you're studying a population. And I always start thinking of Mark McReynolds when I start thinking about gene flow, because he studies those scarlet macaws in Belize. And he doesn't quite know where they nest, and he doesn't quite know where they feed, right? And he's just got binoculars out there, and he's looking at the organisms, but they fly away, and he doesn't know where they go to, but then they come back later. And is it the same bird that came back later? You can't tell unless you've tagged it, right? Gene flow is where you've got populations. So I've got some population of scarlet macaws, and they're either pink or red, feather colors, right? And so, he could go in one season and identify as many scarlet macaws as possible and get an estimation of the genotypic frequencies and then postulate what the the phenotypes, the phenotypic frequencies, postulate what the genotypic frequencies are, and you could start doing some, analysis of this population. What do I think the alleles are that are causing pink and red feathers? But then they all fly away, and they might go mate with somebody in some other population, and then come back the next season with their offspring. So I don't know if that same population was breeding with each other, or if there's some mixing of alleles. We call that mixing of alleles from a different population gene flow, right? Because there could be some other population where, in this case, so the population he was looking at, predominantly they've got pink feathers, right? But they go fly off, there's some scarlet macaw breeding convention someplace else, and another population comes in, and more often in that population that came in, the red was more common, right? So this gene flow kind of gives you the example of it's hard to define what a population is, right? This is not really the population because some individuals from this population are coming in and breeding, right? And so that's going to throw off my calculations. If you've got foreign individuals with a different population allelic frequency coming in and contaminating your sample, right? So this is obviously difficult for organisms that have a wide range, right? But it's difficult when you're talking about human populations too, because we're just typically thinking about, like, historically, what has this population done? Well, that population that's right there now may not be what the population was really, 2 generations ago. Maybe there was a lot of immigrants that came in, and so you're limited by who you can identify as the population, right? So, and then pollen movement too. This is really hard with plants too. How far is that pollen going? I'm doing a population studies with sea squirts right now, actually. So we've taken a bunch of organisms from the yacht harbors locally and isolated DNA out of them and trying to get an idea. We're like sequencing different alleles and just seeing how many alleles are present, what are the frequencies. We're going to go back in the spring, resample the population, and see if the allelic frequencies are changing. Sea squirts just release their eggs and their sperm right into the seawater, though. How far does that sperm float? Does it float all the way down to the next yacht harbor? Does that yacht harbor float all the way up to this one? You have a fertilized egg. How far does that fertilized egg float before it turns into a tadpole and sticks permanently to the rocks and determines what the new population of sea squirts in an area are? This is hard to do, right? Especially if your larva or if your pollen or your sperm or the organism is moving around, right? Most organisms move around and it makes it hard to do genetics on them, right? So most of the time we try and do genetics in isolated populations. If you can find a population of organisms on an island, that's probably good, unless they're birds and they're flying off, but maybe they can only fly so far. Or you can do it in the lab, right? You isolate a population of individuals and study how things are inherited. So, yeah.
I was wondering, 'cause with larger animals, I know that people often tag the animal to know if that's the same animal or whatever. Is there a way to do that genetically, to tag an organism so that when you go back in the spring to study them, you sample the DNA and go, oh, that's our marker that we put in?
Yeah, it's usually illegal to do that.
I'm sorry?
It's usually illegal to do that.
Oh.
Yeah. I could, right? I could do that, right? We can make transgenic animals, and I could, put a transgenic gene in a sea squirt and release eggs and sperm from that transgenic animal out into the ocean, and then I'll know whoever's got my mutant allele that I made probably wasn't a spontaneous mutant, right? If I've got a green fluorescent sea squirt glowing in the yacht harbor and it's a dominant trait, then I know by looking at green flowing sea squirts which ones are going. That's usually illegal, right? You don't want to screw with the population like that. So transgenic organisms sequestered in the lab, it's illegal to release them in the wild because you don't want to mess with it. So the best thing you can do is just genotype individuals, right? And then go re-sequence, their offspring, 2 generations later, and see if anybody has the same sequence that you sequenced in that first generation. So you can track alleles by sequencing naturally occurring alleles, but yeah, you don't want to introduce transgenic organisms into the wild, except for what we do all the time is have been releasing transgenic plants, there are just so many transgenic plants that we're just growing, and you've got transgenic soybeans, and soybean pollen is just floating all over the place, and we know Roundup Ready genes. We know what the transgene are that makes plants resistant to certain pesticides. And so yeah, you can go pick a pea plant and sequence it, and if it's got the transgene in there, you know where that came from, right? So most of the time, if you're working on a model organism, you shouldn't be releasing the transgenics into the wild. But transgenic corn, transgenic tomatoes, transgenic, just all growing them. I wish they would actually force those to be sterile, right? Whatever the transgene effect is doing, if it's making you capable of resisting certain pesticides, I think they should just put in there a gene that makes that plant that has that allele incapable of going through meiosis, right? And we could do that and just make it sterile. But mutations happen, they often get around the sterility issue too. So yeah, we're actually playing a fairly dangerous game with transgenic plants, right? Because we're growing them in huge quantities, and they're spreading their alleles into naturally occurring populations. And what if we lose it? You might have killed off every other genotype for your selected transgenic genotype, right? So it's a good question. I mean, yeah, it's totally possible. Other questions about gene flow? Population genetics in general?
I heard from somewhere, and this might just be a myth, that like as more people can like fly and go to different countries, and it's easier to travel, that like there's more mixing of the alleles between people, and there's going to be like.
Well, if you're traveling to Japan and then you take a Japanese husband and then you come back here, yeah, mixing of alleles.
But what I heard from that was like that everyone's going to end up looking Latino eventually.
Everyone's going to end up looking. Yeah, well, that actually brings up an interesting question. I've always thought, like, what if everyone was allowed to? Not allowed to. I don't condone this. What if every allele in the human population was allowed to mix at random?
Like which one would?
Yeah. What would the consensus amalgam phenotype look like, right? The equivalent of the human mutt, right? I mean,
Go back to Adam.
typically, yeah. I mean, all of the dominant traits are going to be the ones that show up, right?
Yeah.
So probably everyone would be darker skinned than they are now. So yeah, we probably would not see any red-haired people, right? No green-eyed people. Which, I mean, isn't that the glory of things, though? Like, that we have such interesting diversity, right? So, but it is interesting, like, what would be that prototype, for lack of a better word? Yeah. So, yeah.
That's a great segue into my question, which is...
Excellent.
When you talk about natural selection within, and how it affects the genotype, if you were to somehow be able to run such an experiment, wouldn't the outcome be quite affected by the region of the world that you ran the experiment in? So,
Yeah.
like if it was a lot hotter, then the darker, more so than the dark pigment would probably be the more dominant trait that showed up.
Yeah. So that's what I'm saying.
Okay.
If there was no natural selection and just all human alleles were mixing independently of that, what would it look like? Well, dominant traits are going to always dominate the recessives, right? Okay. So the phenotype, I'm saying, what if we just allowed all the alleles to mix and see what the person looked like if you had every allele possible? The most dominant alleles would show up, right? So I guess theoretically, you could think about, it's not even theoretically, this is like in my imagination, right? Take every human trait possible, what is the most dominant version of that trait, put those all together in one individual, what would that individual look like? I don't know, it's just a thought experiment, but you can force populations to adopt certain genotypes and phenotypes. People have been doing this in bacteria. There's a really interesting experiment that just came out last year. This group was breeding bacteria in the lab, and what they ended up doing is they would selectively give them an alternative nucleotide, right? Instead of G, A, T, or C, they had like this chemically aberrant, this different nucleotide. So they grew this population and gave them in their nutrients certain nucleotides. But then they kept increasing the concentration of this rare, weird nucleotide. Now, most of the organisms, their polymerase is not going to recognize that nucleotide. It's not going to get incorporated into their genome, and so you don't see the effect. But sometimes an individual might have a mutation in the polymerase, and it will sometimes introduce it. So we're kind of like selecting for mutations in the DNA polymerase in its active site, the amino acids in its active site that recognize the proper nucleotide. Well, they actually selectively, over multiple generations, just increasing the concentration of the rare nucleotide and decreasing the concentration of the normal nucleotide, they selected for bacteria that recognize an alternative nucleotide. So you have this whole bacterial strain, and they don't put, I forget what it was, they don't put thymine in there, they put, dimethylthymine in there or something like that. It's just a strange variant. But they actually selected for a mutation in the polymerase that recognizes that alternative nucleotide. So you can artificially put selective pressures on populations. And if rare mutations occur, those will be selected for by your criteria, whatever you're selecting for. And then you can force that population to adopt them, right? So yeah, you can do this in artificial breeding situations. So we've been doing, livestock breeding for centuries, and we keep selecting for, cows with smaller horns so that they can get in our pens better, There's very few Texas longhorn bulls anymore because those are really long horns. Like, you can't pack cows in really tightly if they've got those big horns. So kept selecting for ones that are smaller horned, right? And then we still cut them off too. Yeah. Leave you on a really good note. Cutting off cow horns. Yeah. Other questions about population? Then I'm done. We'll talk about sequence alignment, making sequence trees, and how proteins evolve and how we kind of track the change of protein sequences over time next time.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.